Yeah, very good afternoon to everyone. Myself, Dr. K. Jothimani, pursuing postdoctoral fellow in our esteemed research institute under supervision of by Dr. Sri Atati, Research Center for Genetic Engineering at Research Group of Plant Metabolomic Engineering Pathway. I am excited to be here today and share with you some insight on physiological and biochemical analysis of abscisic acid induced drought mitigation of black gram. I will be discussing critical issues that impact us all, the need to significantly increase agricultural food production to meet the demand of a rapidly growing world population. Now let's get started. As we can see in this slide, the world population just increasing, but it is projected to rise more than 1 billion by year of 2030 and by over 2.4 billion in the year of 2050. These figures underscores. Yeah.
Is there any problem with share screen?
Escape first.
Yeah, screen.
From the Zoom.
Yeah, zoom here. Share screen. Share screen.
Yeah. Okay, now you're sharing.
Yeah, sorry for convenience.
Slideshow.
Yeah, okay. Yeah. As you can see in this slide, the world population is not just increasing, but the projected rise more than 1 billion by year of 2030 and by yeah. Full screen. Yeah. Okay. Okay.
Yeah. Not yet. You can slideshow. Press slideshow.
Try the profile.
Udah. Oh, di escape coba diulang lagi, Joti. Escape.
Escape.
Coba.
Share again.
Share ya.
Share.
Udah, aku gambar Bu Sri. Tunggu lama ya. Iya udah.
Sorry, we still start the presentation.
Ketika dia itu share.
Now you can try it on the board.
Nah, ya, sure.
Ya nggak apa-apa nih nobar Pak. Soal antibiotik kecil.
Okay. Yeah, sorry for the inconvenience. As we can see this slide, the world population is not just increasing, but is projected to rise more than 1 billion by the year of 2030 and over 2.4 billion in this year of 2050. These figures undergoes the urgency of our situation. It's estimated the agricultural food production must be increased by 17% by year of 2050. This is not just numerical challenge, but a logistical and technological one as well. In our effort to boost food production, we face numerous challenges on the salinity, fluid, extreme in temperature, heavy metal, radiation, or significant environmental issues. Understanding and mitigating the stress factors crucial for enhancing crop yield and ensuring food security. The water is one of the highest priority in agriculture. However, at least 20% of all developing countries will face water shortage by year of 2030. The drought stress is one of the major limitation of agricultural development, plant growth, and productivity. It's considered being a severe loss of water and a resource. Understanding and mitigating drought stress is essential for improving crop resilience and productivity. And this slide shows that drought stress can lead to morphological, physiological, and biochemical changes of in crop plants. There are some morphological changes of reduced seed germination, small plant size, reduced leaf area, and limited leaf, reduced shoot length. The physiological changes such as reduced photosynthetic pigment rate, increased antioxidant stress, cell wall integrity changes, decreased in stomatal conductance and stomatal closures. Then finally, biochemical changes, declined photochemical efficiency, produced reactive oxygen species, the oxidation and antioxidant defense, reduced chlorophyll content, proline, protein, and high level of ABA accumulation. The black gram mungo is one of the essential rich protein pulse crop belonging to the family Fabaceae. It's mostly cultivated in tropical and subtropical region of arid and semi-South Asia countries, including India, Pakistan, Bangladesh, Nepal, and Thailand. In India, it's an important pulse crop occupying unique position in agriculture, mostly grown in a rain-feed protection system. The black gram containing approximately 25% of protein. These high protein content make it an excellent source of plant-based protein. The fat content black gram is relatively low at about 1.64%. This is make it a healthy option for those looking to maintain a low-fat diet while still obtaining essential nutrients. The black gram is rich carbohydrate with about 60% of dietary fibers and 18.3% of fiber for 100 gram black gram seeds. The abscisic acid is one of the phytohormone, play a crucial role in plant performance under abiotic stress. The abscisic acid and following plus stress hormones. Several studies indicated that ABA could enhance stress tolerance either as a result of its endogenous concentration or through exogenous application. The exogenous ABA beneficial for plant enhanced drought resistance at induced stomatal closure, osmotic adjustment, and increased activity of antioxidant enzymes. It reduced water losses and increased relative water content. And this slide shows the overall objective of this in our study. The first objective is compare the morphological and physiological alteration of 4 varieties of black gram, 1.4, 5, 6, and ADT5, specifically looking at their 7 days old seedling under PEG-induced water deficit stress. Moving on to our second object, we aim to investigate the morphophysiological and biochemical alteration of 2 selected varieties, 1.4 and 6 under drought stress. Our next objective is to find out the physiological to understand how these hormones affect process such as stomatal closure, water uptake, and photosynthetic activity in 1.6 black gram plant under drought condition. The biochemical changes to understand will be provide valuable insights into the molecular mechanism underlying the ABA-mediated stress-tolerance black-gram plants. In this slides, in this figure clearly shows the schematic representation of our objective. The plant material or experimental design. The four variety of plant materials, four variety of black-gram seeds such as 1.4, 5, 6 and ADT5 collected from Tamil Nadu Pulse Research Center, Amban Pudukkottai, Tamil Nadu, India. The PEG-mediated drought stress and 7 days old seedling assess the germination assay. We carefully selected and prepared the seed of our chosen black-gram varieties. We measured several key parameters to assess the plant response. This include the determination of shoot and root length, fresh and dry weight, relative water content, evaluated using the method outlined by Chen et al. They also assess the all parameters using by above mentioned method, standard method. In this next part of in our study is morphological, physiological, and biochemical analysis of 10 days drought stress induced black-gram 1.4 and 6 varieties. And also we analyzed the 10 days drought stress induced with a different concentration of ABA treated in black-gram 1.6 varieties. In this slide shows the result of we evaluate the germination percentage of black-gram varieties under different concentration of 3, 6 and 9% PEG treated black-gram seeds. Each values are present here, represented the average of 3 replication with standard deviation. It shows that the seed germination was significantly affected due to drought stress, the 1.4, 5, 6 and ADT5 varieties. And the germination percentage was decreased with increasing PEG concentration, while the highest decrease was recorded at 9% of PEG treatment in all the varieties. In this slides, we measure the shoot and root length of each black-gram varieties under varying PEG concentration. In this figure shows that shoot and root length decreased while increasing PEG concentration. The shoot and root length showed more reduction in 1.4 at 9% of PEG treatment compared to control and other black-gram varieties. Now we measure the fresh and dry weight of each black-gram varieties under varying PEG concentration. So that compared to control, fresh weight and dry weight of reduced in highest concentration of 9% PEG treatment. The fresh and dry weight was showed more reduction at 1.4 compared with other 3 varieties of black-gram plants. In this graph illustrated the effect of different concentration of PEG 3, 6 and 9% and relative water content, shoot and root proline content for 4 black-gram varieties. The intensity of osmotic stress gradually decreased with relative water content and each varieties when compared to control. The lowest relative water content detected at 9% of PEG treatment in 1.4 compared with other 3 varieties. In this graph, an image shows that effect of different concentration of PEG, MDA content and cell death in 4 black-gram varieties. And the graph illustrated the MDA content both shoot and roots. The MDA content increases with rising PEG concentration among these varieties. 1.6 shows the highest MDA content at 9% of PEG, the suggesting great oxidative damage compared to 1.1 varieties. The image displays the cell death of a seedling. The increase in PEG treatment control, when compared to control, the black ram seedling were germinated and analyzed 3 days, and Evans blue dye used for the analysis of cell death. The result were found that highest concentration of PEG treated root tip increased the blue color formation of compared to control. However, the result was confirmed that increasing the PEG concentration induced high level of cell death in both varieties. This image illustrated the effect of in situ ROS accumulation in 4 black gram varieties using NBT and DAP staining to detect the superoxide and hydrogen peroxide different concentration of PEG treated black gram seedling. The ROS level was increased with PEG treatment when PEG treatment black gram seedling with increasing PEG concentration. The staining of DAP and DAP produced brown color formation of NBT produced DAP produced brown color formation and NBT produced blue color formation of quenching point. These colors indicated that the accumulation of ROS in black gram plants seedling stage. This slide shows the effect of different in the result part of part 2, and the slide shows the effect of different days drought stress and 1.4 and 1.6 black gram plants induced by 5 and 10 days 20 days old plant we treated with 5 and 10 days drought stress. In this result, the effect of drought stress on black gram plants, specifically between 20th to 30th day growth of black gram seedling, that black gram plants. The data shown the data shown is from plants subjected to 5 days and 10 days water stress. Both 1.4 and 1.6 black gram plant stages exhibit significant reduction in shoot and root length, press and dry weight. The impact of is more pronounced with prolonged drought, 10 days, indicating the severity of stress on plant growth and biomass accumulation. In this table, shows that the effect of drought stress photosynthetic pigment 1.4 and 1.6 black gram. The highest reduction of photosynthetic pigment such as chlorophyll A, B, and carotenoid were observed in 1.4 at the 10th day of water withholding plant, then compared to 1.6, then compared to 1.6 and control. This slides illustrated the effect of drought stress on various biochemical contents of 1.4 and 1.6 plant. The relative water content and membrane stability index decreased significantly under drought stress, indicating reduced water content and increased membrane damage 1.4 at the 10th day water withholding plant, then compared to 1.6 and control. The proline content increased under drought stress, showing the plant adaptive response to water deficiency. The total soluble protein content decreased prolonged drought stress, suggesting negative impact of the plant protein synthesis. And this slide shows that the effect of drought stress and enzymatic antioxidant activity such as catalase and superoxide dismantles and melanoidal content always increased under drought stress. In 10th day water withholding 1.6 plant, the total free amino acids was observed in 1.4 at the 10th day water withholding plants compared with 1.6 and control plants. In this figure shows The accumulation of ROS level detected in both 1.4 and 1.6 used by histochemical method. In this histochemical method, the used DAP and the NBT staining provide a visual representation of hydrogen peroxide and superoxide dismutase levels in plant tissues under drought stress condition and control. The ROS level were increased with both black gram varieties compared to the control. Upon the leaf sample, we found that were many conching spots indicating the hydrogen peroxide and superoxide level were high. The high conching points in the brown and blue color conching points indicated the accumulation of ROS level. The highest concentration of hydrogen peroxide and superoxide conching spots were observed in both varieties on the 10th day drought stress induced plant. These figures we observed somatal behavior of 1.4 and 1.6 black gram plants. It indicated that stomatal are closed under stress condition when compared to control. The red color circle represented the closed stomata. The yellow color circle represented the open stomata. From this result, clearly understand about the drought stress increased from 5 days to 10 days. The number of closed stomata increased. In this graph illustrated that drought stress significantly reduced both shoot and root length in the black gram plants. However, treatment with a different concentration of ABA content, abscisic acid, so potential mitigate the effect of this stress with 25 micromolar ABA leading to partially recover in both shoot and root length. In this figure shows that the morphological changes of different concentration ABA treated and under drought stress condition, the morphological changes. In this graph, we observe the fresh and dry weight of black gram under varying abscisic acid concentration. The both fresh and dry weight were increased with 25 micromolar ABA treated plant when compared to drought. In the slide shows the effect of droughters and photosynthetic pigment of different concentration ABA treatment. The photosynthetic pigments such as chlorophyll, AB, and carotenoid were increased. 50 micromolar ABA treated black gram plants when compared to other ABA treatment and drought stress induced black gram plants. In this slide shows the effect of drought stress on electrolyte leakage, proline, and MDA content of different concentration ABA treated with drought induced plant. The electrolyte leakage and MDA content decreased with increasing ABA concentration. The highest decrease recorded at 75 micromolar ABA treated plant when compared to drought. In this slide shows the effect of drought stress on In this slide shows the effect of drought stress on antioxidant enzymes such as catalase, SOD, peroxidase, and APX, different concentration of ABA treatment. The catalase and SOD activity increased with increasing ABA concentration. The highest antioxidant enzyme activity recorded at 75% of 75 micromolar ABA treated black gram plants. The APX and peroxidase content activity also increased with 75 micromolar, 100 micromolar, and 75 micromolar ABA treated black gram plants when compared to control and other concentration. In this slide, in this figure shows in this figure shows the catalase and PO peroxidase enzyme of native page analysis. With the help of non-denaturing page technique, the cat and isozymes pattern was analyzed. The two cat isoforms we detected, in drought and alone, but 3 cat isoforms were detected in ABA treatment. At the high amount of ABA exposed, the intensity of the bands decreased. The POX isoenzymes were analyzed by Page, and we determined that there were 4 isoforms. The 4 isoforms were detected in drought stress treated plant. The isoforms 1, 2, 3 intensity decrease. However, the isoform 2 and 3 were not visualized at control. With other treatment, the intensity of the isoform 2 and 4 was significantly raised at 75% of 75 micromolar ABA dosage. However, it was lower at higher dose of 100 micromolar ABA. In this slide shows the histochemical study of accumulation. The hydrogen peroxide and superoxide accumulation level was increased. Black gram plants under drought and ABA treated plant compared with control. The hydrogen peroxide and superoxide accumulation was visually absorbed by the histochemical method as well as with help of DAP and NBT staining. The high level of quenching spots, the leaf samples was indicated the presence of high level of hydrogen peroxide and superoxide superoxide. The maximum hydrogen peroxide and superoxide quenching spots were observed in 10th day water deficit stress treated plant. The ABA concentration decreased, ROS level, the high level of quenching spots decrease recorded at superoxide 25 micromolar and the 75 micromolar of ABA treated, the high level of ROS reduced in 75 micromolar ABA treated black gram plants. In this figure shows the stomatal activity of water irrigated and drought ABA treated 1.6 black gram plant. The different concentration of ABA treatment were increased the stomatal closure level when compared with ABA treated drought plant and water irrigated plant. In this result, the increasing ABA content increased the guard cells shrinkage, increasing the guard cell shrinkage. The high number of stomatal were closed and guard cells shrinkage were recorded at 100 micromolar of ABA treated black gram plants. In higher concentration, the stomatal guard cell size was also reduced. This is our conclusion and summary of overall the work. The screening of 4 varieties at germination level under PEG-induced drought stress. In this result on germination studies, 4 different varieties of black gram indicated that present seed germination, radical length, total seedling height, and the root shoot ratio decreased with increasing PEG-induced water stress. It's concluded that our previous result, the black gram black gram variety of 1.5 and 6 resistant for drought compared to other two varieties 1.4 and ADT5. In summary of the data presented in this work demonstrated that two black gram varieties were studied, physiological and biochemical, under water deficit condition. The water deficit was reduced the plant growth and biomass, both black gram varieties. In this result also demonstrated that reduction in the photosynthetic pigment, relative water content, membrane stability index, and the total soluble protein under drought stress. We also saw that the long-term drought stress increased the antioxidant activity such as SOD, CAT, and ROS accumulation, superoxide, and hydrogen peroxide level, both black gram varieties. In summary of these physiological and biochemical attributes of the drought stress induced severe damage in black gram plants. The activity of catalase peroxidase significantly higher than that 1.4 and 1.6. In this data demonstrated different concentration of ABA 25, 50, 75, and 100 micromolar impact in the black gram plant, morphological growth rate, biochemical, antioxidant enzyme activities, and the level of hydrogen peroxide and superoxide accumulation. The ABA stimulate the CAT and POX, APX, and SOD enzymatic antioxidant activity. However, 1.6 tolerate drought stress condition by synthesizing extra molecules of radical scavenging enzymes. The isoenzyme band patterns increased with increasing ABA treatment. It is concluded that in our previous result, the exogenous application of abscisic acid treatment slightly increased the drought tolerance mechanism in black gram plants. In this overall percentage, concluded that present study concluded that in 1.6 it's better for drought resistant when compared to 1.4 and other two varieties. In this slide shows the present study of my post-doc research period, the physiological and the biochemical response of cassava PEG-induced in vitro drought stress to select the in vitro selection of drought-resistant cassava plants. We are focusing on some physiological and biochemical analysis to identify the better drought stress-resistant cassava plants. This one is the CRISPR-mediated genome editing drought stress-tolerant cassava. To develop the drought-resistant cassava, in this main aim of the work, investigated the effect of drought stress on genetically edited cassava to examine morphological changes and to assess the physiological response to analyze the molecular alteration of cassava plants. Finally, the goal of this work to select the in vitro screening cassava mediated through PEG identification of drought tolerant genotypes of cassava. Yeah, thank you for providing this opportunity. Thank you, Madam.
Yeah.
Thank you for everyone.
Thank you, Jyoti. Very interesting presentation. I would like to some additional information about Jyoti's work for his fellowship postdoc research. Jyoti has two topics for postdoc. First topic is to establish the method for screening in vitro drought tolerant cassava through in vitro method, and the second topic is how to select screening the plant generated from CRISPR-Cas9 trehalose genome editing. Now we move to next discussion session. We have 15 minutes for discussion. I invite three questions from maybe from chat column, and also you can directly ask to Jyoti.
Satu dulu cari.
Udah di chat ya.
Mungkin dibikin juga enggak.
Ya. Nah. Oh, okay. We have a question at chat column. Yeah. The question is there are two questions. Can abscisic acid be developed as a biomarker for crop breeding drought resistant crop? If it's possible, what is the process of forming the phytohormone based biomarker? And next, second question is, what are the prospects for exogenous abscisic acid and other plant growth regulator to increase plant resistance to drought and field scale? Maybe Jyoti, you can directly answer the question. Or is any question from the audience that can directly ask to Jyoti while we waiting the answer from the chat column?
Ya, yang kedua. Stop share.
Abscisic acid developed as a biomarker. I want to ask this. Abscisic acid developed as a biomarker for breeding crops.
How about the possibility of abscisic acid as a biomarker for breeding drought resistant crop? Is it possible if we use abscisic acid for a marker?
Yeah, it's possible to identify and which genes respond to enhanced the abscisic acid produced gene or abscisic acid suppressed gene. First of all, you identify the abscisic acid. Uh, are you audible?
Or you can move to next question about how is the effect of drought on nodule formation and root morphology of black gram related to change the abscisic acid concentration. Or we can
Are you
invite
audible?
our
Yeah.
question from the audience. You can
Yeah.
raise hand.
What we want to do. Okay, we do have several class. Yeah.
Speak louder. Yeah.
I think
Yeah. Please mention your name and Mbak Rima. Hello, Mbak Lina. Maybe you have a question? Yeah. Okay, while we waiting the response or question from the audience, I have a question, Jyoti. Please, you can some more explain about your experience in Vigna Black Gram research experience and analysis data related to your project, your work in at BRIN about cassava. You develop a screening method for cassava in vitro. Is some the technique you obtain or data related to drought can be applied in develop screening method for in vitro cassava drought.
Yeah. Are you audible?
Yeah. Dapat dengar sakit. Jyoti, can you waiting another question from the audience? Can you explain more about your background experience in Vigna mungo drought tolerant data analysis and also another technique related to your project in cassava drought tolerant? What related data you can apply in the work for cassava?
Yeah, we can apply the cassava for it's similar to yeah, it's audible.
Yeah, we still have 2 or 3 minutes remaining for this session. Sorry for the inconvenience process.
Try to speak some sentences and I will see if we can hear you.
Are you audible?
Audible, iya. Maksudnya?
Audible.
Audible, iya.
Audible?
Yeah,
Kedengeran.
yeah, we can hear your voice. Yeah, you can you can talk about your experience and how to apply your experience to cassava.
Yeah, the plant cassava to induce in vitro drought drought stress tolerant selected with different concentration of PEG to induce the cassava plants and in vitro condition. Uh, to measure the some uh, physiological and biochemical and some molecular characterization. The physiological characterization, number of shoot production, number of root production, and the biochemical characterization such as some enzymatic antioxidant, chlorophyll content, proline, protein, amino acids. The molecular aspects of some uh, drought uh, drought stress tolerant uh, genes are some targeted metabolomic pathways to identify and which some enzymatic antioxidant or uh, uh, which proteins or prolines responds to enhance the drought stress to identify and to enhance the proteins or some enzymatic antioxidant level increases both s- uh, maybe we are used for some uh, other techniques for in vitro or drought selection, some nanomaterial based or CRISPR based
Yeah, yeah. Thank you very much, Jyoti. Interesting presentation. And if the audience need further information, you can contact directly Jyoti or you can contact me. Thank you very much, Jyoti. And for the next session, I invite Dr. Radityo Pangestu as the second presenter.